Assalamu alaikum. I am Dr. Aqsa Perveez, serving as Assistant Professor in the Department of Biochemistry and Biotechnology, the Women's University Multan. Today I will present my work titled Synergistic Chitinolytic Activity of Thaumatin-like Protein and PR1 Protein Implications for Broad-Spectrum Plant Resistance. To understand why this study is important, let us first look at the global challenge posed by fungal pathogens. Fungal pathogens cause significant crop losses globally, threatening food security and economic stability. Annually, up to 30% of global crop yields are lost due to fungal diseases. This represents an estimated $200 billion in economic losses annually, highlighting the urgency for effective interventions. Current chemical fungicides pose environmental and health risks, driving demand for eco-friendly alternatives, focus on biological and genetic solutions. Therefore, sustainable biological solutions are urgently needed. Plants already possess natural defense system, which bring us to pathogenesis related proteins. Plants defend themselves using PR proteins that are activated during pathogen attack and play a key role in systematic acquired resistance. Out of all pathogenesis related proteins, we chose PR1 protein and thaumatin like proteins for our studies. PR1 act mainly in defense signaling, while the thaumatin like protein belongs to PR5 family and shows the strong antifungal activity. However, plant defense usually relies on multiple proteins working together, not individually. So, this idea led us to formulate our research objective. Our objective was to investigate the synergistic interaction between TLP and PR1 proteins using computational molecular approaches. To achieve these objectives, we designed the following in silico methodology. First of all, the sequences of both proteins were retrieved by using the computational data, by using the databases available online. Then physicochemical analysis was performed to find out the basic properties of these proteins. Then by using the some computational tools, the 3D structure of these protein was predicted. Then the interaction study was carried out to find out the interaction of these two proteins with components of fungal cell wall individually, and also then their synergistic action was also studied. Further simulation studies were performed to find out the binding affinity evaluation, and then the synergism action was also studied. So out of this study, we found that the TLP protein has 22 kilodalton molecular weight and PR1 protein has 29 kilodalton. And for further analysis, both proteins show high predicted stability. Moderate hydrophobicity suggests membrane interaction. Isoelectric point value indicates suitability for diverse cellular environment. These proteins support their role as defense protein. After understanding basic properties, we explored their structural features. Secondary structure analysis was carried out. Then the 3D structure model was 3D structure modeling was also performed. The secondary structure analysis showed that TLP proteins predominantly can have beta sheets and random coils, while the PR1 protein has alpha helix, which make that protein the flexible protein, while the TLP proteins is a stable protein. 3D model showed high confidence scores and identified key binding residues. These findings suggested that protein interaction is structurally feasible. Then the docking analysis was performed. Different softwares were used to perform the docking analysis of thaumatin-like protein and PR1 protein with fungal cell wall components that are chitin, chitosan, glucan, and manner. These docking analyses showed the different, here you can see in these pictures, different interacting residues are shown here. For the further study and to check the synergistic action, the protein-protein interaction of TLP and PR1 protein was performed. The protein-protein docking revealed strong interaction with binding energies ranging from 509 to 595 kilocalories per mole, which indicate the stable complex formation. And when the interaction study was performed by using this complex, it showed the enhanced docking score and improved fungal cell wall binding was also observed. The TLP-PR1 complex showed significantly higher affinity for chitin and glucan components compared to individual proteins. The combined action of TLP and PR1 is predicted to lead to more efficient cell wall degradation and membrane permeabilization. TLP and PR1 showed strong synergistic potential. Computational modeling supports stable interactions and also provides blueprint for both protein engineering. This also opens the way for next generation biological fungicides. This research supports us in sustainable agriculture. This helped to reduce resilience on chemical pesticides, environmental friendly disease management, enhance crop resilience, and yield stability. It also supports organic farming practices. So, this was all from my side. For the further detail, you can check the paper published recently in 2025. The reference is given here. If you have any question, you can send an email at aqsa.6497@wgm.edu.pk. Thank you for your attention.